I'd like to introduce myself. My name is Serena McGrogan. I am the College Board's Director for AP Chemistry. And you're asking yourself, am I in the wrong session? Thought I was here for bio. I also used to teach AP Biology. So I taught AP Bio and AP Chem, and I was also an AP Biology reader as well. However, the Educational Testing Service would not let me grade both exams at the same time. It's very difficult to do since the reading takes place in two different locations at two different times. But yes, so I am one of you if you are a biology and chemistry and just science teachers in general. So I do care about ensuring that you have strategies that you feel are effective to strengthen students' understanding of science, whether it's biology or chemistry or physics, etc. However, this particular presentation is going to focus on biology. There are some elements of chemistry folded into it because, as you know, teaching biology, well, used to in the first semester it was all molecular. So there is a lot of chemistry in biology anyway, and it does apply. I want to talk to you, this section focuses on inquiry and the place that it has in the AP Science classroom. And I argue that it has an absolute grand podium stand or setting in the AP Science classroom, because when students discover concepts or an understanding for themselves, it lasts longer, it's retained longer, it's more than just memorizing for the exam. It's, I not only get it, but I'm successful at it. I remembered it for a long time, and if I'm successful at it, then I'm motivated to know just a little bit more, and now I'm interested in it. And if I'm interested in it, I might want to pursue a degree in that field. And if I pursue a degree in that field and I still have the knowledge and I'm really successful at it as well, then I might pursue a career in that field. So for me, inquiry is really important in the AP Science classroom because, again, it helps students retain the information. And I have a great quote here. Blaise Pascal, "We are generally the better persuaded by the reasons we discover ourselves than by those given to us by others." And that's true. When students discover it for themselves, they retain it longer, they understand it better. It's a deeper understanding of the information. And so this particular presentation is going to focus on inquiry and, again, the AP Biology classroom. So I'm going to talk just briefly about what is inquiry, what does it look like. Then we have 4 activities that we're going to do and or look at. One called the Little Red Fish, which everyone should have in front of them. And those of you that came in late, there's an individual right here, the last table on this side, and she'll be able to hand you a red fish in a packet if you need it. So we're going to do an activity called the Little Red Fish. We're going to do one called Genetic Discs, one called the Bugimals, and then another one about DNA fingerprinting. And then I'm going to show you how the activities that I've presented to you today are aligned to the redesigned biology curriculum framework. And then just a little bit about what are some steps you can do to transform the activities you perform in your class into inquiry-based activities. And then afterwards, we'll hold a little Q&A if you have really burning questions that you want to ask. So, what is inquiry? If you look at, many of you as biology teachers are familiar with Latin terms or Greek terms because they're infused in that biology vocabulary that we have to teach. And the word inquire has a very base or root word. Query or inquire means to question. And inquiry is about questioning, but it's about students asking deep questions, not the yes or no questions. It's also about asking testable questions. Not only that, but it's about problem finding and not so much problem solving. So once they find the problem, then they can ask more questions and then they can engage in problem solving. It's also about knowledge creation and not so much knowledge construction. Having them, after they've already constructed their understanding about a concept, then they can apply that to a novel or new situation and then create new knowledge. For example, if I understand how ecosystems work, I can go to my own backyard, do the little grid, it's an AP Enviro lab, do the grid, get all the different species, put it together. There is no paper in a journal somewhere on my backyard. That information is not known to anyone. So I'm going to create it by doing my own inquiry, my own investigation. And it's also about collaboration versus isolation. It's one of those 21st century skills. If you listen to, how many of you attended Tony Wagner's plenary discussion? And he's an amazing speaker, and he said, truly, creativity, innovation, collaboration. Those are really key 21st century skills, and inquiry helps the students develop those skills. And it is powerful. So we're going to start with the little red fish. You have a packet in front of you, and again, for those of you that came in late, there's a lady right here on this last table. And she'll raise her hand. She'll raise her hand so y'all can see. If you need a fish, please see her, and she'll give you one. I'd like you to take it out of the little white envelope and put it in your hand. So take the red fish out of the envelope and put it in your hand. And with the individuals at your table, or you could do that, with the individuals at your table, I'm going to give you 5 minutes. I want you to come up with a question. I'm not even going to give you a question. I want you to ask a question. So give me a minute. I'm trying to pull up my lovely timer. Actually, I'm just going to start now. Go ahead. You got 5 minutes. Come up with some questions, some testable questions about what is happening in your hand with the red fish. It looks as if some of y'all are already testing. All you need to do is come up with a testable question. You got about 2 minutes left. At this time, I'm going to give everybody a format to write a research question. And I'll write it here on the wall, but it is, to what extent does A affect B? And I feel like I'm leaning over. And I would like everyone to write a question in that format. So take the question you already had and put it in the form, to what extent does A affect B? Once you pick them, send them up. No, you don't. Well, yeah, you could come by and say it was the same. Come on over to this side. Come on over. Form a line. Come on over. Just a minute. Sure.
I'm just freezing.
All right. If everyone, as soon as I have a spokesperson for each of the tables, we'll form a line of some sort. Maybe a scatter plot will do. Scatter plot's good. Let's do a slide. And I'm going to tell the individuals, if your question is exactly the same as the person speaking in front of you, you can just come to the microphone and say, that was our question, and then walk out. All right, go ahead.
To what extent does temperature affect curling?
Ours was pretty close, but it was, to what extent does contact temperature affect the rate of curl of the fortune teller fish?
Very close. What is the temperature, or what effect does the temperature of the hand have on the behaviour of the fish?
To what extent does the side of the hand you place the fish affect the fish's behavior on the palm versus the back of the hand?
Similar. To what extent does heat transfer affect the behavior of the redfish?
Kind of similar to one of them. So to what extent does the position you put the fish on the palm of your hand affect how it folds? So if you put it this way or across or diagonal.
Ours was similar to that. We said, to what effect does the position you put it on your body, so we started experimenting with putting it on the back of your hand or further up your arm, affect the movement of the fish.
Since we've had several about the effect of the temperature, I would like to say, what effect does the air vent over a person's hand affect the curling of the fish?
We said, to what extent does the temperature affect the curling of the fish?
Say it again for me.
To what extent does the temperature affect the temperature of the fish?
We had similar questions connecting temperature and curl, but I'm curious about to what extent the moisture on your hand affects the curl.
It's a new one.
We were playing with.
To what extent does good looks affect the curling of your fish?
I like it.
I'm unable to investigate that.
Well, you know, your teenagers, especially your juniors and seniors, are going to definitely have to ask that one. All right, so we've got many here. I typed them down just so you can see a few. There's many. And what's really cool about this particular research question format, I'm going to do this. You ready? What do you like about it? First of all, B rhymes with D, so that makes it really easy. If you have students trying to figure out the independent and dependent variable of a question, you just say, look, take the question and put it in this form. To what extent does A affect B? B is D. B is the dependent variable. A is the independent variable. It's just a cheap, I know I say powerful, but I sometimes say dirty. It's a cheap and dirty way, a shortcut way to teach students, cheap and powerful, that'd be nice. It's a cheap and powerful way to get students to write research questions and then identify the variables. You can even have them write in red and blue. It just sticks out like a sore thumb, and then by the time you're teaching this and they've practiced it, it's down. All right, so I'm going to give you another 5 minutes, and I want you to design a procedure with the stuff that's in this room. And there's a water bottle and some cups right there. I didn't bring anything. Like I said, it's cheap. I didn't bring anything other than those fish. And by the way, those fish are from Oriental Trader, and you get about 144 of them for $4.20. 144 for $4. It's nothing. Oriental Trader. All right, so I'm going to put another 5 minutes on the clock. Come up with a procedure to get data that will answer your question in some shape or form with what we have here in the room. Go. minutes. 2 minutes. All right, we are at time. So if I can have everybody's attention. Literally, that took us about 15 minutes to engage in inquiry with something that cost $4 for 144. Very cheap, and I will show you at the end of this presentation how it is absolutely related to the new curriculum. I'll even give you the learning objective it goes with, and I will even show you the science practices that it is associated with. But what I'd like you to do right now is write down the verbs that you were doing as you were engaging in this process. I had to blank while I was doing the redfish activity. I also had to blank. Just think of words, you know, Bloom's taxonomy, if that helps. I'm not going to say them out loud because that's cheating. Come up with some verbs, Bloom's taxonomy verbs that you were doing as you were engaging in this activity, and then put your redfish away. But keep that list handy. And at your table, you should have two sets or one set. If you're a little table, you have one set. If you're a big table, you have two sets. You've got a little baggie of blue discs and a little baggie of red discs and a little booklet. I would like for you to move everything else out of the way so that either 3 individuals or 2 individuals can play with the red or blue discs in the baggie. For those of you that are in the back, this will probably be a good time just to move a chair to the side of each table so that you can see what's going on. All right, we're going to move quickly. I want to get through all of these activities today. I got this, not the activity because I designed it, but I got the product from SK Boreal. Are anybody familiar with Boreal? I think even the catalog number is on the back of that little booklet there. And it really is not expensive. However, you could make your own, right? In fact, I could probably, you could just draw some squares on a sheet of paper and put big A, big N, little n, and then copy a bunch and cut them out, laminate them, and then the next thing you know, you won't ever have to buy them ever again. So that's what I'm saying, this is cheap. But I didn't have time to do that, so I'm showing you the set that I have. I'd like did you move everything else out of the way? Because I don't want to lose these. Move the water out of the way, move the other stuff out of the way. All right. Have one person dump out the red discs and just kind of spread them out. Have another person off to the other side dump out the blue discs and spread them out. Now what's interesting, in this room alone, I'm going to give you some data. You're the students in the class about to engage in inquiry. And this is how you're really going to have to collaborate. Because I, as the teacher, I'm not going to give you the question. You have to figure it out yourself. I'm going to assume one thing, that you've had regular biology and that you understand phenotype and genotype. I'm going to make that assumption. Can I make that assumption for AP Biology students? I hope I can. Sometimes you got to do a little pre-lesson, but let's assume that. That's the first assumption I'm making in order to make this work. The second assumption I'm making is that every student has a code to what the genes are, and that would be the data table in the front of the booklet. So I'm pretty sure I can make that assumption, just I want you to open it so that you have it there, okay? And it's just a data table. It says XX is this, big N is this, little n is this, big M is this, little m is that. Everybody got it open to the data table? Now, I have 2 and 2 and 2, that's 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. I have 16 mating pairs of individuals. That's good. That's a population of 32. Okay, so 16 males and 16 females in this classroom. This particular gene pool, if you will, because that's exactly what this is, it's a lovely gene pool, has the following traits. I'm not going to list them all. You've got them all right there. I want your particular mating pair, that's like individuals with 3 or 4, I want you to come up with a particular trait that you're really interested in studying and seeing what happens over time, over a series of, excuse me, I just lost my word. Generations, thank you. Over a series of generations, what's going to happen to the gene frequency? I want your group, your mating pair, to decide what you are most concerned about. And then I'm going to ask your I'm going to ask you to meet up with the other mating pair in your table and see if there's some agreement about the gene that you're concerned about, whether it's myopia or tone deafness or is hemophilia on there? Hemophilia and thalassemia is on there. Or maybe you care what color eyes your kids get. Come to a consensus. And then when your table has come to a consensus, I want you to write it. I'm going to flip the chart, and I want you to write it on the board. This gene pool is going to come to a consensus about the gene we are most concerned about and its frequency over a series of generations. So you got about 5 minutes. Has every table had the opportunity? 5, 4, 3, 2, 1. Works every time. Nobody likes that. Okay, I know we're ahead of schedule. It's good. I think we have a clear winner here. That we are going to study baldness. We are. We're going to study baldness. However, and I am going to put a however here, for the sake of time and to make it cheap and powerful, sometimes it's important to kind of rein things in a bit. You know, rein it in. Now, does this particular kid have baldness on the X chromosome? It's separate? Okay. But it's on the X. Yeah. Okay. Let's pretend that. Pretend that's an X with a B on the top of it. Okay, can we all agree that the letter B's are what we're going to look at, but we're going to pretend there's an X there? Okay. I'm going to throw a monkey wrench into the problem in order to make this inquiry, in order to make this group collaborate and work together. And again, I want you to pretend, and it's recessive, right? And it's lower B, and it's on the X. Oh, I need everybody to know that. Huh? What? Capital B. Thank you. Thank you. I don't have them all memorized. Okay. Thank you. Yeah, but so it's a, I thought it was on the X. Is it? Ah, learn something new every day. Okay, so here we go. I'm going to do this. I'm going to throw a monkey wrench in the problem, make it a little bit deeper than before. I'm going to say that in this particular gene pool of individuals, and the particular history of this particular culture or population, baldness is seen as highly favorable sexually for partners. For partners, and I mean that in a biological sense. We're talking about the discs. Highly favorable. And hair, having hair is not favorable, and the females will not mate with individuals who have hair. So I'm just giving you some background. Just giving you some background. And by the way, you could put any monkey wrench into this all you want. You can do selection. You can even put a pre-zygotic, post-zygotic barrier in there. You can all kinds of things. Whatever you want, just make it up. There's all kinds of things. So if I'm giving you that data, this gene pool right here has chosen this particular gene of concern. I want your group to come up with a question. What does this gene pool want to study about baldness, the baldness gene, over a series of generations, knowing that baldness is highly favorable and that hair is not? It's kind of an easy question. This is sort of a guided inquiry, because I'm not giving you the procedure at all. What should our question be? We're all going to focus on the same thing. Someone said, how long will it take before what? Okay, say it again. How long will it take before people with hair die off? Oh, how many generations will occur or go by before the baldness gene is the only, how do we say this? How do we say it? I like it. Until we have a homogeneous baldness population. I like it. But don't give us the answer yet. Don't give us the answer yet. Not yet. Not yet. Do you want to throw another monkey wrench into it? Do you want to give the kids a little more data? You can go as deep with this as you want. You can make it surface and make it easy, or you can add all kind of other epigenetic, all kind of effects that you want to. Even protein, even transcription regulators, you can throw those in there as well. You can say, you know what, in the food diet, for whatever reason, zombie apocalypse occurred, and the food that's available now, kids will love that, especially the junior, senior boys, zombie apocalypse, and the food that is available does not have the protein that is required to transcribe that particular protein that decreases the the hold that the follicle has on the hair. And you could throw that in there and then see what happens. There's all kinds of things you can do with this. Does everybody see that? And then from then on, students can ask that question, how many generations before X? Or they can ask, well, how much, I'm trying to come up with on myself. Let's throw some Hardy-Weinberg equilibrium in there. Yeah, thank you. How long will it take before the allelic frequencies change drastically? You know, I think it's about the same, but one is looking at it in a macroscopic way and one is looking at it in a microscopic or calculation way. Either way. So to me, there's so many things that you can do with just those little discs sitting right there in your hands. In fact, you don't even have to focus on one gene. You can have the kids focus on a series of genes and make their own little groups of individuals with only 4 characteristics and just say, all right, play around with it, gene pool. Tell me what happens after 20 years. And see what they come up with. And then they're going to have all kinds of data that will lead to even more questions, which will lead them to doing even more population genetics experiments, etc. Okay, so I don't have more time to spend on this, and I'd like you to put the blue discs back in the one bag that says, I think it says male, female, and put the red disc back in the other. And then have one person from your table gather the bags and give them to me. Yes, and the booklet. Thank you. So the discs and the booklet. Thank you so much. At this time, make sure you have that envelope at your table handy. There should be an envelope. It may say Fort Bend ISD and it may just say a number on it, but it is an envelope. And this is by far one of my favorite inquiry activities. I love it. You're going to still want to make sure that you have space at your table. So you want, and this time, you're really going to want to clear off space. So I'm going to ask you put the papers that are yours, water bottles, etc., under the table and clear off the entire table and make sure you have an envelope between 3 individuals or 2, depending on how many you have at your table. Or 4, that's right, we had some more people come in. All right, this is hands down one of my favorite activities to do because its implications are far-reaching, and it's kind of like all of Big Idea 1 in an envelope right there. Maybe not, but you can translate it across all of Big Idea 1. So in your envelope, you have, I already cut them out for you. You have the little bugamals. If you don't want to call them bugamals, you can call them whatever you want. And the kids can actually call these whatever they want. Let's just say, for sake of time, these are bugamals. I already cut them out for you and put them in an envelope. You should also have a list or a word splash of evolution terms that are already colored and cut out in your envelope as well. I would like you to separate the terms and the bugamals because they got mixed in the shuffle in my bag. So just kind of separate them out. I promise you it's important.
Face up or face down?
Face up. You're going to want to look at your screen. All right, so I hope that you've got it separated out. Everybody ready? And you don't need the string. You can just pretend the string is there. I just couldn't travel with scissors. I'm loaded with string, but not scissors to cut it. Here we go. You have a population in front of you. And you can see all of the bugimals in front of you. I would like you, as an evolutionary biologist, to make up a question about those particular bugimals. And I want you to pretend, because this is imaginary. Part of inquiry is creating and recognizing patterns. But I want you to pretend that you're loaded with data about pre-zygotic, post-zygotic barriers, about temporal isolation, mechanical isolation. You can even see some of it in front of you with the organisms that are right there, all their structural features. Pretend you have all that data. As evolutionary biologists, I want you to come up with a question about this particular species. You got about 5 minutes. Come up with a question. And guys, if I can have everybody's attention, I want to give you a short little footnote. I want you to relegate your question to the following parameter. Pretend that you could find the answer or understanding of data pertaining to your question by the development of an evolutionary tree or cladogram, whatever you want to call it. So just pretend that that is going to give you insight to your research focus. All right, if I can have your attention. 5, 4, 3, 8, 7, 2, 1, and 0. Okay. I'm actually going to have you focus on a question just for the sake of time. You can do this all you want, but for the sake of time, there was a research question that I heard, and it was about the change in locomotion or locomotive appendages. Is that what you guys said?
Yeah, change in locomotion and means of.
Okay, change in means of locomotion over time. And they feel that if they have, all the data in front of them, and again, this is data you're going to come up with, you're going to make it up. I would like everyone in this group to devise an evolutionary tree that shows the change in locomotive means over time, and then at the different branches or separation, at the different points of speciation, I would like you to insert an evolutionary term if you think it applies. And if you need more terms, I think I have extra. So if you think, pre-zygotic barriers occurred twice, you can just come up and get another little index card and insert it there. And I'm going to give you about 15 minutes because this is fun. I think it's fun. If I can have your attention, we have about 6 minutes left. That was good. Okay. Normally I have to count, but you made it easy for me. Give me just a second here. I need one person at your table, or who was using, the group of you that was using the contents of the envelope, to put it back in the envelope, please. And I am going to move quickly through this activity, the next one. So if you'll just take a little bit of time, put the contents back in the envelope, including the little index cards and string, just kind of shove it in there. And then have one person from your table bring it forward. And then pull out the packet that says AutoRad or DNA fingerprinting. So just make sure you have that packet in front of you. It probably has typos all over it. All right. Please pull out that packet that says AutoRad or DNA fingerprinting. I just want to go over it just a little bit. Again, I'm running out of time, but I do want to make sure that I touch on this and then also close out, the session. It's important to have closure. All right, how many of you have seen the Arizona State Biology Project website? I think it's Arizona State Biology Project. None? One? Show of hands. I just want to see a few. It's an amazing website. I'm going to link you to it right now. There is one particular activity that you can have the students do as just a guided activity through it with the actual questions and everything right there. It's called the Blackett Family DNA Activity. For DNA fingerprinting, I love it. The kids love it. But you can also make it inquiry. You don't have to use this simulation. So I want to show you in your packet what you actually have. And a way to take this particular simulation that guides them through what is an AutoRad, what is DNA fingerprinting, and who is the Blackett DNA family, and how can I prove that they're actually family. That's what this is. But you can actually get rid of everything in the front and simply show them an AutoRad. And say, all right, guys, you may not have heard it before. You may have heard the word DNA fingerprinting in a court case or in the newspaper. What is that? What does that look like? All right, it looks like one of these. And one of these is called an AutoRad. It stands for autoradiography. And the process is called RFLP, or restriction fragment length polymorphism. But all that information is just kind of, it's really gobbledygook to the students. They're not really listening to you when you say that. But you want, part of inquiry is showing them data and letting them explore the data and find patterns for themselves. So what you can do, and again, we're running out of time, so I'm not going to do it with you, but you've got the information right here on the slides, and all you have to do is Google search the biology project from Arizona, arizona.edu, and it'll bring you right here, and you can do the tutorial all by yourself. But this is what I do. I simply talk about what DNA fingerprinting is. I simply show them what an AutoRad is, and give me just a second. I'm going to come up here to the wall. I tell them what a ladder is, so a DNA ladder, showing all the different sizes of fragments of DNA. So take a piece of DNA, cut it with an enzyme, and you get that many fragments. And they are of known size. So it's really important to tell them about the data. And the ladders are throughout this lovely gel. Because if I had a ruler way over there, and I'm standing right here, and you want to know how tall I was, would that ruler be right? Would you be able to tell how tall I was exactly if a ruler was way over there? No, it'd be nice to have one right here next to me. So when you want to know the size of a fragment of DNA, it's nice to have a ruler or a DNA ladder, a handy laboratory ladder right here next to all the different bands. So it's throughout the gel. Again, I'm not going to teach you the whole thing. But anyway, you've got some unrelated individuals over here, and it's real important to include unrelated individuals on a DNA fingerprint. It's a control of sorts. Then you've got mom, dad, brother, sister, grandma, grandpa, and some other unrelated individuals. And what you can do is, once you've just given them that little bit of information and telling them that a piece of DNA cut with a particular enzyme, and that enzyme is cut at specific sequences, like AAGC or TTCA, depending on your sequence, you will get so many fragments of DNA. And if you have a similar sequence as someone else, I'm being guided back to the microphone. If your sequence is similar, and I mean very similar, to someone else's, then when I cut your sequence and that someone else's sequence, you're going to end up with the same size fragments because it cut in the same place. And once they have that little bit of information, you can decide just how much information you want to give the students. Then you give them an actual autorad. You can say, oh, here's, if you turn to the second page, you'll see something titled D1S7 and D2S44. You can give them both, you can give them one. D1S7 is chromosome 1, section 7, and D2S44 is chromosome 2, section 44. Those are actually two markers that are unique to individuals and families. So when you cut pieces of DNA within these two sections of chromosomes, unless your sequence is the same, then you're going to have different size fragments than the person standing next to you. Again, you decide how much information you want to give the students. And then, again, remember, parts of inquiry is about identifying patterns and then coming up with additional questions. You've got unrelated individuals on here. And just a spoiler alert, some of the unrelated individuals look like they have the same size bands as the ones that are supposedly in a different family, which is really great. It leads to more questions. You can ask the students, all right, I'd like you to tell me who is mom, who is dad, who's grandma, who's grandpa, and prove it. And then ask additional questions. Who's unrelated? How do you know? Prove it. Justifying claims with evidence. And again, you can decide, you can give them, if you turn to the next page, you have two more chromosomes. Chromosome 4, section 139, and chromosome 10, section 28. You can give them all 4 autorads and say, all right, give them one autorad and say, all right, prove it with this, and then really mess with them. Give them the 3 more autorads and say, are you sure? These are 3 more. You just proved it with 1 chromosome. I'm positive I share, humans share what, 99% of their DNA? Well, I'm pretty sure I got a gene on chromosome whatever, 4. It's probably the same as yours or yours. So just using 1 autorad is not enough. Let them come up with that. Give them 1 and then give them 2 more. And then say, prove it again. Let's see if you can justify your claims with evidence. So that is that's the DNA Blackett family project, and I would urge you, because I didn't have time to really explain it fully, we're running out of time, I urge you to go to the biology project, do the simulation yourself. It's really easy. Do the project, the activity yourself, and then you decide how much information you're going to give students up front and what questions they might ask. But at this time, I would like to finish up the presentation. So some of the things that you need to ask yourself about the activities you are already doing in your class. Well, ask yourself, is it inquiry? What is inquiry? First of all, inquiry activities focus students' inquiry on questions that are challenging, that are debatable, like the DNA Blackett family activity. There's a lot that's debatable there. A student might say, that fragment is 100 kilobase pairs, and the other student goes, I disagree with you. From the way I'm reading the little ladder, that's 98. Very debatable. And difficult to solve, like the evolutionary tree, the cladogram. None of you here had the same tree. It was not the same answer from any of you. Expecting students to apply concepts to new situations, so not just looking at the protein sequence, the amino acid sequence of the beta hemoglobin chain of chimpanzees, orangutans, and humans, but just giving them maybe some false data pertaining to those buggles. It's a new situation. Structuring lessons so that students have opportunities to work with peers, learning collaboratively, which all of you did here. None of you worked in isolation. I hope you didn't. And then expecting students to figure out patterns. And I would like to say the activities I showed you today were all about patterns, maybe not the redfish. but the other ones were. And then teaching students how to think and to engage in problem-solving skills. That's how you know it's inquiry. And it doesn't have to have every one of those pieces, but many of them count. The science practices that are in the biology curriculum framework, and I'm going to focus on that as I said before, they're really a series of verbs to represent, to justify, to solve, to describe, to model, to predict. And I would ask, show of hands, did you do any of that in the last hour? Show of hands. Keep them up. Then we engaged in the science practices, and we're engaging in inquiry. And if you do something like, say, if you already have activities that are similar to this, or want to use things that are similar to this, or like this, or this, you can be sure you did it, you realize those were your actions, and the students are engaging in those actions as well. Here are the learning objectives of the biology curriculum framework that align with these particular activities. So the little red fish, explain the connection between the sequence and the subcomponents of biological polymer and its properties. Well, the little red fish is not a biological polymer, but it is a polymer nonetheless, and it still translates to understanding structure and function. It's sodium polyacrylate, I believe, is the name of the biomolecule, and they use it, I'm not biomolecule, the name of the molecule, and it's used in diapers to absorb water. And I'm cheating now. I really shouldn't have told you. I really wanted you to go home and look it up. Genetic disks, 1.3, apply mathematical methods to data from a real or simulated population to predict what will happen to the population in the future. Wouldn't you agree that's what we were about to do? We ran out of time. The Bugamals, refine scientific models and questions about the effect of complex biotic and abiotic interactions on biological systems, from cells to organisms to populations to ecosystems. You could take that Bugamal activity and pull it across the big ideas all year long, if you wanted to. And then lastly, DNA fingerprinting. So construct an explanation using visual representations, and I definitely argue an autorad is a visual representation about how DNA and chromosomes is transmitted to the next generation, and then you can have students feed in their explanations, mitosis and meiosis followed by fertilization, and that activity directly applies. So it's all about aligning what you're doing to the new curriculum framework. So how can you take what you're already doing and make it inquiry? And this is just an algorithm. It's not the whole thing. It's a cheap way to take your activities and make them inquiry. Have students explore without you being involved. Again, you can give them a little 5-minute speech like I did up here. I didn't tell you very much, but I let you explore on your own. And then have them question, which I also asked you to do. It's important that they inquire. And then have them design, either design a model, a pattern, a representation, or a procedure. And then have them collect data to justify the extent to which their model really aligns or helps predict a particular pattern, or provides answer or support or refutes their hypothesis to a question. And then collaborate to understand. What I would have done after you developed your cladograms or evolutionary trees is as you get up and look at everybody else's. Talk to the different groups. How did you come up with that? Man, mine was really different. I disagree. Listen to them, collaborate, and discuss. And that's a really good way. I call it a 5-step system, a really good way to transform what you're already doing in your classroom to inquiry. So for those of you that did not receive handouts, this is my email address, smcgrogan@collegeboard.org. I do admonish you to go on and fill out your ranking or rating of this presentation and to provide feedback. And also to email me in case you have additional questions or you want, a PDF of whatever it is that I've shown you, or just other information. I do have, we are out of time, but I don't think there's any session after that. So if we want to hold a little 5-minute, 10-minute Q&A, we'll start over here.
Will you email us this presentation?
The steering committee of the conference has asked me for my presentation, and they're going to post it online. So you don't need, and it's a big file with all the animations. It would destroy your inbox, I promise. So they're going to post it online. Online, Email me when you get a chance, and then I'll tell you where it is, because I don't even know that. Yeah, probably the AP Annual Conference website. But if not, just email me and I'll tell you. Thank you, everybody, and I hope you have an amazing conference.